Hey guys, so today we're going to start kind of a big one. Um, Let me just get to the beginning of this. This is RNA and protein synthesis. This is probably going to take up a couple of lectures, just to give you the heads up. This is very, very important. This is possibly the most important stuff that we talk about all year as far as like its impact on biology. So DNA is the genetic material of cells. The sequence of nucleotide bases in the strands of DNA carries some sort of a code. In order for that code to work, the cell must be able to understand it. So what exactly do the bases code for? So the A's, the T's, the G's, and the C's. That's what we're talking about. Where is the cell's decoding system? So we're going to start off talking about RNA. And how does RNA differ from DNA? There are three important differences between DNA and RNA. The sugar in RNA is ribose instead of deoxyribose. So DNA is deoxyribonucleic acid, RNA is ribonucleic acid. Two, RNA is generally single-stranded and not double-stranded. And three, RNA contains uracil in place of thymine. So there are no T's in RNA. We replace those with U's. So in DNA, we have A, C, G, and T. In RNA, we have A, C, G, and U. Each nucleotide in both DNA and RNA is made up of 5-carbon sugar, a phosphate group, and a nitrogenous base. There are three important differences between RNA and DNA. We already talked about those. So similarly, the cell uses DNA master plan to prepare RNA blueprints. The DNA molecule stays safely in the cell's nucleus. If you remember, that's where the DNA is found, in the nucleus, while RNA molecules go to the protein building sites in cytoplasm. Those are the ribosomes. Going way back to the beginning of the year, we talked about what the different organelles do. And the DNA stays in the nucleus, and the proteins are made on the ribosomes, which are out in the cytoplasm. You can think of the RNA molecule as a disposable copy of a segment of DNA, kind of like the working copy of a single gene. RNA has many functions, but most RNA molecules are involved in protein synthesis only. RNA controls the assembly of amino acids into proteins. Each type of RNA molecule specializes in a different aspect of the job, and we're going to talk about each one. So the three main types of RNA are messenger RNA, ribosomal RNA, and transfer RNA. We're really going to focus more on messenger and transfer RNA, which we call mRNA and tRNA. Ribosomal RNA basically just makes up the ribosome itself. So the messenger RNA is the molecule that carries copies of the instructions known as messenger RNA. They carry the information from the DNA to other parts of the cell. So they act as a messenger. So they're the ones who take the DNA, or the code from the DNA, I should say, from the nucleus out to the protein synthesis area, which is the ribosome. Proteins are assembled on ribosomes, small organelles composed of 2 subunits. The ribosomal RNA is one of those subunits. And then transfer RNA is how we kind of build the protein. So when a protein is built, a transfer RNA, tRNA molecule, transfers each amino acid to the ribosome as specified by the coded messages in the mRNA. So you'll kind of see how this works. So how does the cell make RNA? In transcription, segments of DNA serve as templates to produce complementary RNA molecules. So the first part of this process is called transcription. Most of the work of making RNA takes place during transcription. During transcription, segments of DNA serve as a template to produce complementary RNA molecules. So we're going to make RNA strands from the DNA. The base sequences of the transcribed RNA complement the base sequences of the template DNA. In eukaryotes, we'll skip over prokaryotes. In eukaryotes, RNA is produced in the cell's nucleus and then moves out to the cytoplasm to play a role in the production of proteins. Our focus will be on transcription in eukaryotic cells. Okay, so this is inside the nucleus, and you have an enzyme called RNA polymerase, which is very similar to DNA polymerase. And this RNA polymerase is going to put together the messenger RNA molecule. RNA polymerase binds to the DNA during transcription and separates the DNA strands. Then it uses one strand of DNA as a template from which to assemble nucleotides into a complementary strand of RNA. So what is the genetic code and how is it read? The genetic code is read 3 letters at a time so that each word, that's what we're going to call it, is 3 bases long and corresponds to a single amino acid. The first step in decoding genetic messages is to transcribe a nucleotide base from DNA to RNA. This transcribed information contains a code for making proteins. The proteins are made by joining amino acids together into long chains called polypeptides. As many as 20 different amino acids are commonly found in polypeptides. So basically what they're showing here is if you join up these amino acids, okay, by a peptide bond, you make this polypeptide chain. The specific amino acids in a polypeptide and the order in which they are joined determine the properties of different proteins. Once again, this is really important. We are made up of proteins. We're just tons of protein. Therefore, making these proteins correctly is very important. The sequence of amino acids influences the shape of the protein, which in turn determines its function. RNA contains 4 different bases: adenine, cytosine, guanine, and uracil, so A, C, G, and U. These bases form a language or genetic code with just 4 letters. So each 3-letter word in mRNA is known as a codon. Okay, a codon consists of 3 consecutive bases that specify a single amino acid to be added to the polypeptide chain. So these 3 letters here are a codon. The middle 3 letters here are a codon, and then these 3 letters over here are also a codon. So this codon is AUG, this codon is AAC, and this codon is UCU. Because there are 4 different bases in RNA, there are 64 3-base codons, 4 times 4 times 4, in the genetic code. The circular table shows the amino acid to which each of the 64 codons corresponds. To read a codon, start at the middle of the circle and move outward. So we'll take this. We'll say AAA. Okay. You would start with this A and then come and choose the A out of here. So A, and then I said the last one was A. That would be lysine. That would be the amino acid that would be coded for. We'll say GCU. All right, here's G, C, and then U. So that would be alanine. CCA, C, C, A, that would code for proline. So what role does the ribosome play in assembling proteins? Ribosomes use the sequence of codons in messenger RNA to assemble amino acids into polypeptide chains. I think we're going to stop here, and then we'll pick it up in the next video.